All right, in this section, we're going to talk about the plasma membrane specifically and how substances are able to cross the plasma membrane. So after this section, you should be able to describe the structures of the plasma membrane and list the types of molecules found in the membrane. You should be able to distinguish between the different processes of diffusion, osmosis, and facilitated transport. Explain how tonicity relates to the direction of water movement across a membrane, and compare passive transport and active transport mechanisms, and then finally summarize how eukaryotic cells move large molecules across a plasma membrane. So, the plasma membrane. As we stated in the previous section, it is common to all cells. It's going to be the outer boundary of the cell, and it's going to regulate the movement of substances into and out of the cell. That phospholipid bilayer that we mentioned previously when we talked about phospholipids are going to separate the extracellular fluid from the cytoplasm. Inside of that, or embedded in that phospholipid bilayer, we're also going to find proteins, cholesterol, and carbohydrates that'll help give it the qualities of a fluid mosaic. So the fluid mosaic model is just how we describe it. It's because it's sort of made up of this constantly moving fluid bilayer. The plasma membrane is composed of this phospholipid bilayer, and those phospholipids, as we mentioned, have a polar phosphate head, which is going to be hydrophilic. It's going to orient itself to the extracellular water and the water found on the inside of the cell in the cytoplasm. The nonpolar lipid tails are hydrophobic, and they're going to orient themselves inward. This is how we sort of end up with a sandwich of polar phosphate heads on the outside and nonpolar lipid tails on the inside. So that's how the cell is going to, or how those membranes are going to orient themselves. So the plasma membrane, that phospholipid bilayer with proteins, they're going to be attached and embedded within that phospholipid bilayer. And like I said, when those phospholipids are placed in water, they sort of form a spherical bilayer. They're going to form that sort of circular shape that gives the cell its characteristic circular appearance. So there you go. So this depiction here of the plasma membrane is often referred to as the fluid mosaic model, and that's because the various proteins, lipids, glycoproteins, and carbohydrates which are embedded inside that plasma membrane have sort of the unique ability to be very fluid. They can move sort of in and out. You can kind of think of this as boats or buoys in the surface of the ocean. Some of these buoys are going to be anchored to the surface below. So we have some proteins that are going to be anchored to the cytoskeleton. They are not going to move, but the lipid bilayer can actually move and flow very easily past these embedded proteins. This embedded bilayer can also flow and move easily past each other. So they can kind of slide back and forth and move all around in that plane. In the same way that water molecules in the ocean can slide past each other. There are also boats that can zip along and move throughout that layer. If we had a buoy that was not anchored, it could kind of float and bob along and move in any of those directions in that plane. So the mobility and the flexibility that the phospholipids provide gives such freedom and flow of movement that it has the appearance of being kind of fluid in nature. In fact, at body temperature, the plasma membrane has the consistency of olive oil, so it's very slippery and has very fluid in its movement. The phospholipid bilayer has several unique characteristics that we've kind of already mentioned, but are already covered, but are worth mentioning again. Like I said, that phospholipid bilayer is the external surface lined with hydrophilic polar heads, and the cytoplasmic side is also going to be lined with those hydrophilic polar heads. The nonpolar hydrophobic fatty acid tails are going to be sort of sandwiched in between. The double bond in the fatty acid tail is going to prevent close packing, which is going to help contribute to the fluidity of the membrane. The greater concentration of the unsaturated fatty acids residues, the more fluid the structure of that cell membrane. And like I said, these monolayers, so the single layer, can actually move and slide past the layer below. So it's kind of like if you had a sandwich with too much jelly, the pieces of bread could move and slide past each other very, very quickly. Some membrane proteins can act as channels. So we've already mentioned that there are proteins embedded in the plasma membrane, and each of these proteins sort of have their own purpose. Some of those membrane proteins can act as channels. They're going to allow some things in while keeping other substances out. They're going to be small hydrophobic substances can pass freely through the phospholipid bilayer. So things like gases like oxygen and carbon dioxide, anything that's very small and non-charged can move very quickly and rapidly through that phospholipid bilayer, which is fantastic. We're going to see later on when we talk about cellular respiration how very important it is to have lots and lots of oxygen available to our cells and the ability to quickly remove carbon dioxide. And since those can pass freely through the plasma membrane, they're going to diffuse really very rapidly, and we're going to be able to move those in and out. Ions, so any molecule that is charged, is not going to pass through freely. It's going to need a little bit of help. So any large or charged molecule is going to need some help passing through. Typically, we're going to use a channel protein that's going to sort of create a bridge that will allow those molecules to move through. Water molecules are going to have a very specific channel protein called an aquaporin, which is going to allow water to move and pass through the cell. So even though there's water on this side of the membrane, water on this side of the membrane, water is not going to cross through this hydrophobic desert, if you will. This region right here is very hydrophobic. Water is not going to be able to pass through, not without the help of a channel protein. So there are two types of movement across the plasma membrane. There's passive transport. Passive transport is going to be movement across the membrane that does not require any energy. This will be things like simple diffusion, facilitated diffusion, osmosis. And then there's active transport. Active transport is when we have movement across the membrane that requires energy. That's where we get the active from. Active transport is going to, so some examples of this would be active transport, endocytosis, exocytosis. All of these are going to require the input of energy in order to get molecules across the plasma membrane. So we're going to actually talk through some of these different types of movement, starting with diffusion. Simple diffusion is just the movement following a concentration gradient. The end result is an equal distribution between the two areas. So what we have initially over here is we have a higher concentration of these little purple particles and these water molecules on this side of the plasma membrane and a lower concentration over here. Over time, we're going to see an equal distribution. We're going to see an equal movement. We're going to see that we have sort of the same concentration of water and particles on this side as we have on this side. So what happens is over time, we're going to reach equilibrium. Net movement has stopped. We have just as many particles going from the right to left as we have going left to right. There are a lot of things that can affect simple diffusion, things like temperature, pressure, electrical currents, molecular size. All of these are going to affect the rate of diffusion. And so when we're crossing the plasma membrane, the things that can diffuse would be things like small and uncharged molecules. Carbon dioxide, oxygen gas, those can move very freely across the plasma membrane, and they're always going to move through diffusion, moving from areas of higher concentration to areas of lower concentration until equilibrium is reached. So there you go, diffusion. Diffusion is just the random movement of molecules from an area of higher concentration to an area of lower concentration. And they're going to continue to move until they are equally distributed. This is, of course, passive movement. There's no energy that is required to move those molecules from an area of high concentration to low concentration. Molecules can move in both directions. So if you see in this slide, we're going to see that molecules are moving from right to left and left to right and right to left and left to right. They're moving in both directions. But what we're going to see is that more molecules are moving right to left than left to right until we reach equilibrium. So until we have an equal concentration all throughout the room, we're going to see that movement. So no energy is going to be required, and that once we reach equilibrium, the same number of molecules move into and out of the cell. You can kind of think of this, if any of you have ever used a diffuser or like a scented mister. what you'll see is when you spray a burst of scent, you're going to have a very high concentration of scent in that area right around the can. But those molecules, those scent molecules, are going to slowly move from that area of high concentration around the can until they spread out and fill the whole room. They're going to diffuse throughout the room. Now, there's not going to be any input of energy. You're not going to add energy to that. You're not going to be intentionally like trying to blow or move those molecules around the room. If there's no air currents in the room, those molecules are still going to eventually disperse and spread throughout the room, moving from an area of high concentration to an area of low concentration. So that's diffusion, just the movement down a concentration gradient from high to low concentration. Osmosis is very similar to diffusion, but it's a very specific form of diffusion. The study of osmosis focuses primarily on the movement of the solvent, typically water, across a semi-permeable membrane. Semi-permeable membrane just means that it's going to allow some, but not all molecules to flow freely across. So in this case, the semi-permeability refers to the ability of water molecules to flow freely across the membrane, but the solutes cannot. So if we go back to this diagram, it would be like if water could move across the membrane, but these little salt particles could not. So on one side of the membrane is the solute solute concentration is high, but the water concentration is low. On the other side of the membrane, the solute concentration is low, but the water concentration is high. The water is always going to be moving from an area of high concentration to an area of low concentration. And there are several different things that are several different terms that we want to define when we are talking about osmosis. And that's going to be when we're referencing salt concentration or the strength of a solution. So normally body fluids are going to be isotonic to cells, meaning that the fluids found in your bodies, your interstitial fluids, your blood is going to have the same concentration as your cells. So your cells are not going to change drastically in size. However, what would happen? Does anybody know what would happen if we were to inject pure water into an IV? So if you've ever gotten an IV at a hospital, you'll know that an IV is usually to keep the body hydrated and to keep your blood pressure up because you're keeping your blood volume up. This is done by injecting an IV solution or injecting a saline solution into the bloodstream. We don't inject pure water into the bloodstream. Pure water has a higher water concentration and a lower salt concentration than our red blood cells. If you were to inject pure water into the bloodstream, you would have a hypotonic solution. This would cause your red blood cells to swell up and maybe even burst or lysis. The term for red blood cells that would swell up and burst is hemolysis or hemolysis. You can pronounce it either way. And you'll see this happen frequently if you work in the healthcare field. If you've ever worked with blood samples, it is possible for those blood cells to burst. Of course, that's outside of the body in a centrifuge or whatever. But that's the bursting of those red blood cells is the hemolysis in a hypotonic solution. A hypertonic solution would have more solutes than your cells. So the cells are going to shrink, undergo crenation. Osmotic pressure is going to drive this. So here's an example of what that looks like. In the laboratory, cells are going to be typically placed in an isotonic solution. That prefix iso means the same as, and tonic or tonicity refers to the strength of the solution. So an isotonic solution basically means the same strength. So in an isotonic solution, the solute concentration and the water concentration, both inside and outside of the cell, are totally equal. So we're going to have just as much water flowing into the cell as we have flowing out of the cell. There's going to be no net gain or loss of water. The solute concentration of red blood cells is roughly 0.9%. So any IV solution is typically going to have a 0.9% of salt, and it's 0.9% NaCl solution. This tonicity keeps the red blood cells happy and healthy. Solutions that have a concentration lower than that of the red blood cells are, of course, hypotonic. Hypo meaning less than, and tonicity, again, referring to the strength of the solution. If a cell is placed into the hypotonic solution, it's going to cause all of the water to rush into the cell, Any solution with a salt concentration lower than 0.9% is hypotonic to red blood cells. Animal cells laced with a hypotonic solution will swell and maybe even burst. And we're going to pause right there, and I'll see you guys in just a moment to finish up.